made out of glucoses, they don't count as sugars. And so the benefit does not detect starch, but it does test for monosaccharides and disaccharides. The Benedict's test is a little, slightly more complicated to run. You put in some drops of the Benedict's reagent, which is this nice, pretty kind of sky blue colored stuff here. The handout will tell you exactly how many drops. I don't remember, to be honest, but put in some drops of that. And then you have to boil the tube for a few minutes. So you have to get out the water bath, get out this hot plate here, and get a beaker. Oh yeah, a few tips here. Use the deionized water, which is in these cardboards right here, for your boiling. Just turn the spade here and fill it up with water. Well, don't fill it up, maybe about halfway should be fine. If you use the water out of the sink, when you boil it, you get these crusty calcium deposits. For some reason, our water here is really, really, really high in minerals. And once you boil them on there, they're almost impossible to get off. And so you'll be here until midnight trying to scrub off those magnesium salts or whatever they are. Anyway, so boil it in deionized water. And these little round things at the bottom are called boiling beads. They help give you a more smooth boiling. If you don't have them in there, it goes when it's boiling. I'm not sure why. Why doesn't our pot of water on the stove do that? I guess because the pot of water is iron or aluminum and this is glass. But anyway, it'll boil more smoothly if you have these little beads in there. But don't throw the beads away when you're done. When you're finished with the experiment, it's time to clean up. Dump the water out down the sink, and this thing will catch the beads for you, and then you can put them back. Actually, put that right here when you're done. And then wash the beaker out and dry it with paper towel. Put it back. Oh yeah, so what do you do with the Benedict's? Well, okay, so with the Benedict's, after you've boiled your tube for a couple of minutes with the Benedict's mixed in, if you have sugars, the Benedict's will turn kind of a brighter color, usually red, if you have a lot of sugars in there. If there are none, then the Benedict's reagent doesn't change color, and it stays that nice sky blue color. And again, you can have something in between. If you get green or yellow or orange, you have some sugars, but not a whole bunch. All right, now, unfortunately, the Benedict's reagent has, I think, copper ions in it, which is where it gets blue from. And it's not the type of thing we're be chucking down into the ecosystem. So for the Benedict's reagent, when you're done with it, when you have your test tubes, dump them down in here. The iodine stuff is safe enough to flush that down. Let's see. Yes, that's the gist of it. Just two more quick announcements. Rolando stepped down for a second, but he's thinking about starting a biological engineering club. It's a field he's interested in. Can't seem to find a good club that really exists on campus. So that might be something you might be interested in. You might want to talk to him about maybe getting a club going. And the other thing is, I'm going to tell you about your midterms. So while you're working today, I'll pass them back for you to review. You should look over how I add up the points on every page. And the bottom right-hand corner of the page is the total points lost for that page. That's what the circle one is at the bottom. So I make sure that I did the math correct. I'm usually pretty good about it, but I do make some mistakes. Make sure all your points subtracted out give you the correct point total. I have 100 on top. If you don't think you were graded fairly, if you think you put down the right answer and I didn't give you credit for it, I will not get angry with you questioning me. Just come out here and I'll discuss it. I can't guarantee that I'll give you the points, but I'll at least discuss it. When you're finished with it, though, I want to recollect it from you. Unlike most teachers, you do not get to keep these. Review them today, but when you take the exit quiz, I'll get them back. If you want to look at them again, that's fine. I can bring them to other lab periods, or you can come to office hours and I'll show them to you again. Now, some people say, well, how will I study for the final? Well, for the final exam, I'm not going to use these questions. I'm going to use, well, who knows what I'm going to use for the final exam? I mentioned on the first day. Review questions, right. In your textbook, at the end of each chapter are some review questions. Those are what are going to appear in the final exam, not these questions. All right, yes, that's it. Do you have any questions? All right, go ahead.
Before we start,
Oh yeah, I mentioned that. I saw you. Do you want to add anything to it?
no.
Okay, so that's Rolando. He's going to try to maybe get a bio.
Oh no, actually, well yeah, actually I just want to make a bio club in general, but not like related to specifically for medical, so like molecular biology, cell biology, botany, zoology, so anything that doesn't have to do with medical.
It's kind of funny. We have a physics club, an engineering club, and a chemical engineering club, but we don't seem to have a biology club other than the agreement club. So, more power for you. All right. I guess that's it. So start in. If you have a question on the procedure,